Greetings, Bio 2010 students. We will be jumping into the Brooker text, beginning with Chapter 7 on photosynthesis. I'll begin this chapter with a brief overview of photosynthesis itself. We will be marching our way through the chapter as it's outlined, with the exception of section 7.5, which covers the variations in photosynthesis. You will not be held accountable for that information. So let's start by talking about the big picture of photosynthesis itself. So really, the takeaway from this whole chapter is you should be able to understand how is it that plants and other photosynthetic organisms, such as cyanobacteria, how is it that they're able to produce organic matter, such as glucose and oxygen, when they utilize CO2 and water as their starting products. Now, if you think back to chapter 6, there we were taking glucose in the presence of oxygen and breaking that down to make a lot of ATP. The byproduct of that as well was CO2 and water. And so plants and, as mentioned, cyanobacteria, they're able to do the inverse of that. They're taking CO2 and water, and with the help of the reactions that take place in the chloroplast, they're able to convert that into oxygen and glucose. You can see that overall reaction here. Now, when we're talking about photosynthesis, it's important to differentiate between autotrophs and heterotrophs. Now, autotrophs implies that one is able to make their own food, auto implying self, whereas heterotrophs imply that we have to get our energy by ingesting and digesting food. So again, autotrophs are able to make their own food. They're able to take inorganic carbon, such as CO2, and make that into their own glucose supplies. Heterotrophs, on the other hand, we are unable to do that. And so we have to take in our glucose usually by eating things like plants, and then we will break that down into CO2 and water. Again, where does the energy come from in order to create this food? So for an autotroph where they're taking CO2 and water and converting that into glucose, again, if we are building something up that's going to take energy, and where does that energy come from? Well, for the most part, what we will be talking about is using the sun as a source of energy. And we just did this in lab a few weeks ago. Another possibility is that this could come from other chemical reactions, which is what we sometimes find in the deep thermal vents in the ocean. Heterotrophs, on the other hand, as we're breaking down things like glucose, that's how we're going to generate our energy. So this all fits together very nicely. Again, if you start at the top of this figure with chapter 6, we were taking glucose and its byproducts. We were taking glucose in the presence of oxygen, and we were able to break that down into CO2 and water, a byproduct of which was a lot of energy. However, if we're going to build something up, if we're going to build a glucose molecule that's going to take energy, where does that energy come from? It's going to come from the light. Okay, before we go any further, let's do a quick concept check. I want you to think about which type of organisms carry out cell respiration. Is it A, heterotrophs, B, autotrophs, or C, both? Go ahead and pause the video and answer the question on your own. Okay, hopefully you selected C, both. Whether we're talking about autotrophs or heterotrophs, they're all able to still break down glucose. It's just only the autotrophs who are able to take the inorganic carbon, the CO2 and water, and convert that into something like glucose for themselves to utilize. All right, and we can see that displayed here. If we're talking about photosynthesis and the ability to make glucose, that is true only for our plants and cyanobacteria, but then everything, whether we're talking about plants or animals, bacteria, etc., they all are going to undergo cellular respiration, which is one of the main ways that we make a lot of our ATP, which is necessary for all of our other chemical reactions in the body. All right, so let's talk a little bit about the actual structure of a plant cell and how this is a bit different from our own cells. So one of the main things that you'll notice is that plants have a cell wall, whereas most animal cells do not have a cell wall. Bacteria also have a cell wall. This is really important for providing structure, structural integrity, for being able to resist things like turgor pressure. When the plant cell fills with a lot of water, the cell is not going to burst. That's going to be really important for maintaining the plant itself. The other thing you should notice is a lot of students mistakenly think that plant cells do not have mitochondria. They indeed do. You'll see they have other membrane-bound organelles, such as peroxisomes. However, one of their other really unique structures is the presence of a chloroplast. So if we are talking about photosynthesis itself, that's going to take place solely in the chloroplast. And I like to zoom in on this. So here we've got the chloroplast itself, and we've got an inner and outer membrane, which should sound reminiscent of what we talked about with regards to the mitochondria. And then within the inner membrane, we have these stacks of thylakoids, and the space in between the thylakoids is called the thylakoid lumen, and a stack of those is called a grana. So here again, if we take a single plant cell, and each of these green circles here is going to be a different chloroplast. So the plant cell itself is full of numerous chloroplasts. I've included this other image of a chloroplast because there's a couple of features here that aren't as well depicted in our textbook. One thing I like to point out is that chloroplasts have their own chromosomal DNA, just like ribosomes do as well. And so when we're talking about a plant cell, they're going to have their nuclear DNA, mitochondrial DNA, as well as the DNA found in the chloroplast. The other thing I think that is depicted a little bit more easily here to see is between the outer and inner membrane is this inner membrane space, and then all this space on the inside that includes your thylakoids and the granum and the grana and etc. This is called the stroma, and that'll be really important when we're talking about the specifics of photosynthesis later on. Going back to the images taken from your textbook, again, there's going to be 3 sets of membranes, which just means we have a lot of compartmentalization within a chloroplast. We have the outer membrane and the inner membrane, as we've already discussed, and then those thylakoids are going to be made up of membranes as well. So those are all places where if we were able to zoom in, we would see an actual phospholipid bilayer. So all the photosynthetic reactions, this whole chapter can be summarized in this image here. So again, you now are more familiar with the structure of a plant cell. We've zoomed in within just a chloroplast itself, and we're looking at one set of grana, one set of thylakoid stacks. And really all we are doing is taking a water molecule, and we're going to break open that water molecule by a process called photolysis and take energy from the sun and infuse that into the electrons from the water, a byproduct of which is going to be oxygen. The other main byproduct of these reactions is the synthesis of ATP and NADPH. Now, NADPH is not something we've talked about yet, but it is very similar to something we have talked about, which is NADH. If you think back to the role of NADH in cell respiration, the role there was simply to be a high energy electron carrier, and that's the exact same role of NADPH. So NADPH and ATP are then able to take inorganic carbon, CO2 and water, and they're able to synthesize sugars as a byproduct such as glucose. So we have two distinct sets of reactions. We've got the light reactions, which is really breaking the water molecule open, producing oxygen, ATP, and NADPH. And then that energy is utilized to take something like CO2 and build a much larger molecule, such as glucose, C6H12O6, with the energy obtained from ADP and NADPH to make things like sugars. So again, if you think about our reaction that we did in chapter 6, we said that the delta G for this reaction was negative, which means it's exergonic. Energy is going to be given off when we do this reaction. So if we're going to do the opposite of that reaction, which is chapter 7, taking CO2 and water and making glucose, we're going to have to put energy into the system. And so this is an endergonic reaction. And you can see that looking at the delta G's, one is a negative 686 kcals, and now the synthesis of glucose is going to be the opposite of that. It's going to take 686 kcals per mole. So one thing I want you to be able to answer is what is oxidized and what is reduced in this reaction. So take a minute and make sure that you can answer that question. So this is a brief overview of section 7.1. We talked about the overall reaction. We talked about where this is all happening within the plant cell itself. And then we talked about the two stages in which we're talking about the conversion of light energy into chemical energy, which includes both light reactions as well as the Calvin synthesis.